Hi, loves. I wanted to just do a quick overview of heredity and genetics. This PowerPoint will be on Schoology, so if you have any questions or would like to review it, just let me know. If you look here on this title slide, there is a video that is an overview of how genetics and heredity are connected, so feel free to check that out as well. So heredity and genetics are kind of paired together because genetics is the study of your DNA and the genes that are contained on your DNA. Those are segments that code for traits like your height, your skin color, color blindness, any trait that we might have, or that is passed down to us. Heredity is the study of how organisms pass on traits from one generation to the next. So whereas you have people in your family, or this may even be you, you have traits that make you unique. Your eye color, skin color, hair color, your height, whether you can roll your tongue or not, all those things are traits. But you may have gotten those traits from your father's side of the family, or you may have gotten those traits from your mother's side of the family, and heredity is the study of how we pass those traits down and which traits become passed down. So let's go back a little bit to mitosis. So mitosis was a cell division process to help you grow and heal. There is a cell division process that's necessary for genetics and heredity, and that would be meiosis. Meiosis is simply how do we make those sex cells, the sperm and egg cells, they're going to carry the chromosomes from your parents to you. So you will get 23 chromosomes from your mom, and you'll get 23 chromosomes from your dad, and they're contained in those gametes, and those gametes are created by meiosis. And then when they combine during fertilization, then you get all 46 of your chromosomes. So you get 23 from mom, 23 from dad. That's where you get the whole 23andMe, those 23 pairs. So once we make a you, we have to be able to know what type of traits you are going to have. When you do something like 23andMe, they often do karyotypes where they lay out all the chromosomes. So you have chromosome number 1. And the reason why there's 2 here is because you got 1 from mom and 1 from dad. Same thing for chromosome 2. You get 1 from mom and 1 from dad. Chromosomes 1 through 22, we call those autosomes. Those are the chromosomes that are going to determine what your body is going to be like. Are you going to have blonde hair? Are you going to have blue eyes? Are you going to have brown hair? Are you going to have brown eyes? Are you going to have a hitchhiker's thumb? Are you going to have a widow's peak? So 1 through 22 determine all of the characteristics of your body or the traits of your body. The last set of chromosomes are your sex chromosomes, the X and Y, and these determine gender in mammals. So normally they are set aside, and right here we would have a male because we have an X chromosome and a Y chromosome. If it was a female, they would both be the same size. So how do we take the information that's on those chromosomes and actually turn it into a trait that you see, like hair color? Well, this is a process called protein synthesis. So in every cell, you have the nucleus, and in the nucleus is in that DNA. And you don't want to copy down every single piece of information and turn it into a trait. So you only want to take down the specific information that you want. And that is done through transcription, where you take the DNA and turn it into essentially like a shortened message. And that shortened message leaves the nucleus and goes to a ribosome, and the ribosome makes the protein. Here is another view of that process. So here you have a cell, and in the nucleus there are those chromosomes. And chromosomes are just wound up DNA. So the DNA will unwind, and a message will get copied. Here you see that process. You've got the DNA, it gets unwound a little bit, a copy is made using an enzyme. That enzyme makes something called a messenger RNA. That messenger RNA leaves, and then it goes to a ribosome, and the ribosome can make the amino acid, can make a protein chain. So how do we track these traits or those proteins that get passed down from mother to offspring or father to offspring? You have something called a pedigree, and that's what this is. This is a pedigree. It's a colorful pedigree with pictures of people, and I'll show you a different type that we normally look at in science. So here we have John, our focus person, and this is John's karyotype. On his karyotype has the information for what type of traits he will have. So how do you know what type of trait you're going to get? How do you know if you're going to get blue eyes from your mother and your father, or if you're going to get brown eyes? So let's look at some of the information on chromosomes that determine which trait we get. So remember, a gene is a piece of DNA that tells the cell what type of protein to make, and you got that information from your parents. Each gene has a specific area on it that has that gene location. We call those alleles. And alleles can either be dominant or recessive. So here you have an allele or a location for wrinkles. And these are wrinkles on pea plants. So if you have both the same, so we have wrinkled and wrinkled, we call that homozygous. So this person would definitely code for, or this pea would definitely code for wrinkles. Over here, you have wrinkled and round, and we call this heterozygous. In certain genetic situations, this would definitely code for the wrinkled because we have a dominant trait. To get the homozygous, the recessive trait, sometimes you have to have both, so round and round, but we'll get to that in a second. So with heredity and genetics, like I said, you can have two types. You can have Mendelian genetics or non-Mendelian genetics. Mendelian genetics is either or traits. You either have the dominant trait or the recessive trait. Dominant is normally shown with uppercase letters. So here we see a dominant purple flower. And so it's purple because it has both dominants. Over here, you also see a purple flower, but it's got a dominant trait and a recessive trait. And remember, so one of these Fs is on one chromosome and the other one is on the other chromosome for maybe number 1 or number 2. The reason why it's purple is because these are Mendelian genetics and you only have either or. You have the dominant trait or the recessive trait. And here for these flowers, the recessive trait is white. So you see the only way that you can get white is lowercase f, lowercase f. Now with non-Mendelian genetics, you normally have a blending of traits, and this is what we more often see. Nobody's just, this or that trait. And a lot of times multiple genes, even on different chromosomes, code for certain traits. So sometimes it takes 2 or 3 genes to actually code for a trait. With this, sometimes you have a red flower mixed with a white flower and you get a pink flower. That's what we talk about, that blending of traits. So where over here we didn't get a light purple flower, we either had purple or white. That's what we mean by either or. Over here with non-Mendelian genetics, you have a red flower mixed with a white flower and you get pink. And then oftentimes if you mix 2 pinks, then you'll see the white and the red show back up. Hi, so let's look at how we blend heredity and genetics together, and we often do this on a pedigree. So let's look at sickle cell anemia. It is a recessive disorder, which means that you have to have a lowercase letter and a lowercase letter. So mom would give you the recessive disorder and dad would give you the recessive disorder. And sickle cell disease is carried on chromosome 11. So if you were to look on a karyotype, it would be held on this chromosome. So here we have Arlene and George, and Arlene, you can see she has lowercase h, and that means she carries that recessive disorder. And on pedigrees, when someone carries a disorder, we often shade in their circle or their box. And circles represent girls, boys represent boxes. So Arlene and George, as you can see, were married and had offspring. And here they have one offspring, Tom, who has sickle cell, Sam, who does not have the sickle cell disorder, Wilma, who does have the sickle cell disorder, and Anne, who does not. You also see that they have married. Thomas married Sandra, and they had offspring. And then Anne married Michael, and they also had offspring. Anne's offspring, Carla, also appears to have the sickle cell disease. So oftentimes we can look at the genetics of, people like George or Sam or Michael and try to figure out what their genetics are so we can maybe figure out if Daniel and Alan are either carriers or if they don't have a chance of passing on sickle cell to their offspring. When you used to do Punnett squares back in 7th grade, this is one of those times that you would use it for. So if I took, let's say, let's look at George and Arlene and see if we can figure out and make sure that what we see represented here is correct. So we have Arlene, lowercase h, and then we have George. He's an uppercase H and an lowercase h. And one of the things that we really don't get to talk about is meiosis and the process of how we actually separate those chromosomes from each other during that gamete formation and that type of cell division. So you can often use the Punnett square to check and see if a pedigree makes sense, or if, a lot of times when people are doing paternity testing, they might see something in their family tree that just doesn't make sense, and so they might have paternity testing or maternity testing done to make sure that their parents are really their parents, or the offspring are actually their offspring. Those are those whole separated at birth, and people who find out they're related after being apart for years and years. So we have first combination would be a heterozygous when you have an uppercase and lowercase. Another probability would be another heterozygous. And then a third one would be the homozygous recessive, where that offspring would have sickle cell disease. And I'm proposing that George is heterozygous. Okay? Because we have offspring that do not have sickle cell disease. So Sam is most likely heterozygous as well. He got uppercase H from his dad, and he got a lowercase h from mom. He would have been one of these combinations. And then Wilma got the lowercase h from her dad and a lowercase h from her mom, being one of these combinations. Now you can use the Punnett square to check and make sure, what if George was homozygous dominant? So let's erase all this. Okay, so here we have Arlene. We know she's definitely homozygous recessive because the only way to have sickle cell disease is to be homozygous recessive. But let's say if George was homozygous dominant, meaning he has uppercase H and uppercase H. So all of their offspring would be heterozygous. And since we have Wilma and Tom that are have the recessive disorder, we know that George has to have a lowercase h. Okay, so George can't be this option. All right. If you have any questions about heredity and genetics, please feel free to reach out to me. This is going to be posted on Schoology, and I will have a couple other resources there for you. Feel free to reach out to me anytime. I really miss you guys. Hope you have a great day. Bye.